Uh, thanks to you both, to you, the three previous speakers, because really they were very, very, very good and really helped me to present what is happening in chronic spontaneous urticaria according to different examples that we have. As you know, we have as main objective to obtain complete relief of symptoms, no hives, no angioedema, no itch, as fast as possible. And we need to treat our patients as long if it's necessary, looking for the minimum effective drug. This is real practice and is not necessarily what is shown in the clinical trials. Also, there is another objective for chronic spontaneous urticaria that we know from the ligelizumab studies, that if we reach complete response, we can achieve almost 100% of the benefits in the quality of life, sleep, activity, or work from our patients. And these are, as clinicians, we are involved, all of us, in clinical trials, and always is the last objective when we develop a drug according to the different diseases. Nowadays, this is the algorithm that we just have, and the only biologic that we have that was omalizumab, that is omalizumab and is viable. It was developed, as it was mentioned, directly in the phase 3 with two parallel studies that were the two Asterias in people who were refractory to the anti-H1, and another third study, Glacial. It was directly a phase 3 study because it was a previous indication on asthma. And here you can see after 12 weeks how the obtention of the urticaria activity score 0 was compared to the placebo. It's an extraordinary drug approved in the 2014, first by the EMA and after by the FDA, and now we know how to manage it after 10 years ago. But this trial was just 6 months trial compared to placebo, and it was not reflecting the real world practice when we treat our patient of CSU. We learned from this Asteria that really we need for some patients more than 3 months in order to complete control the disease. And coming from some observational study developed during the real world practice, exactly from the AWARE, and focus on the European countries, urticariologists' expertise, we can see that after 2 years, even the people were well treated with omalizumab in green, and also the amount of cyclosporine was less and the amount of montelukast was less. These columns show people that really felt them bad control, not completely control according to UCT. And thanks to omalizumab, thanks to all the UCARE network, thanks to Marcus Maurer, thanks to the Charité, thanks to all the urticariologists in the world, we pushed and we put the urticaria in the center of the translational medicine. Then, because there is an unmet need, different clinical trials were developed and different triggers were addressed, IL-1, PGD2, and CS5, with no success. And really, there were different trials, very well conducted, and Peter shown a slide with the ligelizumab, who really spent all the phases from the development of the clinical trial till phase 3. Phase 3 completely developed and demonstrated, which was very effective compared to placebo. But now, after all this complex development, will not be available in the market for CSU, as it will not be available in the phase 3 benralizumab or tezepelumab, who really are drugs very effective for other indications that really develop a good program but failed to be absolutely perfect compared to the patients treated with placebo. I explain you this because it's not so easy, and especially for the patients, they should know that this complexity of the development of the clinical trials that involve many stakeholders, pharmaceutical companies, etc., sometimes it fails. And now this long list was reduced to this. And I will explain you just 3 examples from the new treatments that are addressed now and probably will have in the future. One, One of them is addressed to this complex inflammatory infiltrate that commonly goes through the wheel after the muscle activated and release mediators and chemo attract mainly lymphocytes, eosinophiles, etc. And is dupilumab. Dupilumab, you know, it has the indication for atopic dermatitis, also for prurigo, nodularis, and because part of the inflammatory infiltrate in the wheel pathogenesis is stage 2 and mediated by the IG, then there is a development of dupilumab for CSU. The difference is that in this case, we went directly to a phase 3 with a very few number of the patients included. And look, here there is an example. We've demonstrated, or has been demonstrated in the CUPID-1 with a very few, no more than 100 patients, that Dupi is more effective than placebo, but it was not extremely better. It was better, but not extremely better compared with the people that were previously treated and failed to omalizumab. What happened? It was approved in Japan directly. It has approved in the Arabic countries. But when it went to the FDA said, we need more patients. And then the CUPID-2, the CUPID-1 was created, and they've created Sanofi, the CUPID-3, and after a long period or longer period of development for the phase 3, they obtain also benefit in the patients that were naive to omalizumab. And now they went to the agencies. Let's see if they are approved. Is applied to the EMA and will be in the future. 2 other drugs, completely different mechanism of action. We know that if we selectively control the brutontarosine kinase, inhibiting the brutontarosine kinase with a very selective way, covalent and also irreversible, as remibrutinib, it works, we can manage and we can interfere in the expression, intracellular expression, then the expression of the high affinity Ig receptor, not just in the muscle, but also interfering the production of the immunoglobulins. It's a different complex mechanism of action. Then it both a thing which is new when we face the drug and we express and we show the drugs to the agencies, to the pharmaceutical companies. Then here you have just an image from the phase 2. It has been described a phase 2 previously by doctors. We have 5 doses reaching from 10 milligrams once a day to 100 milligrams twice a day. And we see how the different doses benefit in the reduction of the Urticaria activity score 7, which was the primary endpoint. Coming from the phase 2, they've choose the dose going to the phase 3. And how it was explained before, there are two parallel phase 3 study with around 300 patients per study. And here you can see the core phase, which is compared to placebo. And these patients that were treated with placebo, when they go in the open phase during 1 year in order to assess safety, because in the trials we should assess efficacy and safety, especially for the BDKs, then such people who were in placebo get a benefit from the drug. See, very, very similar in both studies. And here, when they go, is the similar, but is the way how you obtain an urticaria, how they obtain US 0. Then, according with the safety, is important just this image, because for the ITBDK we have this challenge, and the challenge is to be safe. And to be safe, and this is just the image when we, in the long phase during 1 year, 52 weeks, in the phase 2, using 100 milligrams twice a day, the drug was demonstrated safe, but also should be demonstrated in the phase 3, also during the 52 weeks, etc. And finally, the other revolution that will be in CSU is to modify, try to deplete trigger the leader cell that induce the wheel, which is a muscle. This image came from the slides that commonly Marcus shown, and you know that there are some anti-KIT that really interfere with the stem cell factor, and this anti-KIT, if they interfere with the stem cell factor, then they deplete the muscles from the dermis And we know that in CSU and inducible urticaria, there is an increase of these mast cells in the dermis. We are depleting them. Now in this phase for barzolvolimab, which is the first anti-KIT and the more advanced anti-KIT development, we have just the results from the phase 2. And you can see we have a comparison between 75, 150, and 300 milligrams because we check the appropriate dose to go to the phase 3. And you can see that 150 and 300 milligrams has a benefit enormous compared to placebo after 12 weeks. And normally the trials for CSU follow the same patterns. We always use the primary endpoint tend to be each in the United States, but also the urticaria activity score 7 in Europe. And also we check at 12 weeks, 6 months, and the different times. And we look for the efficacy. And here you can see how many people obtain well-controlled urticaria activity score 7 lambda disease or 0, which is the goal, our challenge, obtain 0. And you can see the differences between compared with placebo with barzolvolimab, this anti-KIT. This reminds a lot what happened initially with omalizumab. Is a little bit different what we have and with Remi or with Dupi. Remi is very good also, but it's different. And here, and the challenge now that we have nowadays is because we have the background for omalizumab, if these drugs will work also in people who were refractory to OMA. There are not many patients completely refractory to OMA, but there are at least 45% of patients that need to for controlling the disease to updose twice or 3 times the dose of omalizumab. There are a good number of partial responders. Then this is an image about the increased number of mast cells in induced by the TEM test by cold. And also we have another challenge. We have CSU, we have 2 challenges for urticaria is to obtain the indication for the inducible urticaria, which are very rare, but impair the quality of life of the patients a lot. And for barzolvolimab, when we have this, when we really study cold urticaria or symptomatic dermographism, this initial study in phase 2 looks very well in all the patients with cold urticaria. They reduce the temp test, the minimal temperature that induced the wilt, which is normally the mean 16 degrees Celsius, and really just after 1 shot, it reduced in all of the patients the dermographism, the threshold that induce wilt when you friction your skin, and also this will correlate with the delta-Y and the control of the disease. This will correlate with the serum tryptase, which is decreased, and the increase of the plasma stimulant cell factor, and etc. Then nowadays, there is long list that really was reduced to 6, which are Dupi, remibrutinib, rilzabrutinib, and barzolvolimab, and some others which are in phase 2. For example, this anti-JAK is in phase 2. There are others which are really in development. It has been reduced. But and I can guarantee that in the next future, this long list reduced will be, look, again, 15 drugs in development, especially different anti-KITs and different anti-BDKs and other potential anti-IgE therapy and even anti-high affinity IgE receptor therapy, etc. But the main message is we need new treatment for CSU, for sure. We need new treatments in order to guarantee the indication in children. We need the indication for inducible urticaria. And this presentation want to show you the patients that how difficult it is to develop a new drug, how frustrating it is when a very good drug as ligelizumab will not arrive to the market, and how evidence and how helpful is when the patients accept to be involved in a trial, because this will help for them and also for the general knowledge of the disease that they are suffering. Then thank you all for your attention. Catherine, we can end. Thank you so much.
Thank you, Anna. Great presentation.